Welcome to Question of the Week. This is a Facebook Live session where I take on your science faith questions Mondays, 4 p.m. Pacific Standard Time, 7 p.m. for those of you on the East Coast. This week's question comes from my friend Steve McCrae, who, by the way, is the organizer for the Great Debate Community, which is a Google Hangout group. So if you're really interested in in-depth and vigorous science faith discussions, I would invite you to check out the Great Debate Community on Google Hangout. And I would also invite Steve in the comments to put a link to, for people who are interested in learning more about the Great Debate Community. But Steve asks a question, actually a couple of questions. The first is, are mitochondria bacteria? And the second question is, what do I think about the autogenic origin of bacteria, check that, of mitochondria and eukaryotic cells? And these two questions are intertwined and relate to a larger question, and that is there a strong evolutionary model? Is there a strong case that can be made from an evolutionary perspective for the origin of eukaryotic cells and mitochondria and the endosymbiotic hypothesis? So I'm going to be taking on those questions and more in just a minute. But before I do that, I like to ask those of you who are watching this broadcast to take a moment and to check in the comment section. Let us know who you are and where you're watching from. This is really very important information for us to have as we plan future broadcasts, but it's also a great way for me to get to know who you are, to know a little bit more about you, and I really do want to get to know all of those people who are interested in this particular broadcast and in science faith discussions. And in fact, the last time that we did one of these broadcasts, which was a few weeks ago now, September 11th, I believe was the date, a number of you took the time to check in, including my old friend from Spencer, West Virginia, who I went to junior high and high school with, Jesse Starcher. So I was really grateful to hear from Jesse from Spencer, West Virginia. Aaron Perez from Trinidad and Tobago. Mitch Milan checked in from Kauai on the island of Hawaii. I met Mitch and his wife Sharon a number of years ago when I was doing some outreach events and vacationing on the island of Kauai. In fact, they had my wife Amy and I over for dinner, and we had an evening hike on the beautiful island of Kauai. So it was great to hear from Mitch. And then finally, Nigel Nyaker checked in from Durban, South Africa. So thank you so much for all of you who took the time, again, to check in and let us know who you are and where you're watching from. Also, take a moment and react to the broadcast with the like button. The more likes that we get, the greater the visibility this broadcast will receive in the Facebook feed. So you're really helping us out quite a bit by liking the broadcast. And if you don't like the broadcast, or if you have ways in which you think we can improve the broadcast, please offer your comments in the comment section. We're interested in hearing what you have to think. And then finally, if you want to offer your thoughts or your perspective on the endosymbiotic hypothesis, on the origin of mitochondria, either from an evolutionary or a creation model, or just want to offer some general thoughts on the creation-evolution debate, please feel free to do so in the comment section. Even if your perspective is different from mine, I want to hear from you. I want to hear what you have to say. It's very important to me to foster an environment on my Facebook pages, both my public figure and my personal Facebook pages, where people can discuss science faith issues, very important issues, without any kind of censorship, without any kind of restriction or constraint. Okay, without any further ado, let's go ahead and take a look at the questions that Steve McCrae raised. And as part of that, the broader question, is the endosymbiotic hypothesis a valid evolutionary model for the origin of mitochondria and hence the origin of eukaryotic cells? And we'll start off by saying this, that one of the key transitions in the history of life on Earth is the origin of what are called eukaryotic cells. Now, it's not really quite clear to me from the fossil record exactly when eukaryotic cells originate. There is some controversy as to the timing for the origin of eukaryotic cells. Typically, you'll see people argue that it was roughly about 2 billion years ago in Earth's history based on the fossil record. There's some geochemical evidence that maybe eukaryotic cells would have originated a little bit earlier than that, as far back as perhaps 2.8 billion years ago. But this is a key transition in the history of life on Earth because complex organisms, animals, plants, fungi, are made up of eukaryotic cells. And so the origin of these complex organisms could not have happened until eukaryotic cells would have originated. Now, prior to the origin of eukaryotic cells, the cellular life on Earth, which was single-celled organisms, were made up of prokaryotic cells. These are cells that constitute bacteria and archaea as two separate domains of life. These are cells that are about a micron in size, typically speaking, though some may be larger. And these cells are characterized by having a cell envelope, a plasma membrane and a cell wall, maybe an additional outer membrane structure, but there's no internal structures observable with a microscope for prokaryotic cells. Now, there's evidence that the molecular constituents of prokaryotic cells are actually organized within the interior of prokaryotic cells, but there's no visible evidence of subcellular structures. Now, in contradistinction, eukaryotic cells are much larger in size, 20 to 30 microns typically speaking, and they're characterized by having a large membrane-bound spherical structure in the interior called the nucleus, as well as an internal membrane system called the endoplasmic reticulum that gives way to a Golgi apparatus, as well as membrane-bound structures called organelles, such as mitochondria, if these cells are animal or plant cells. And the question is, how did eukaryotic cells originate? And one of the ideas that is currently considered to be the preeminent explanation from an evolutionary standpoint would be something called the endosymbiont hypothesis. This is an idea that traces its beginnings back to the early 1900s, where a Russian botanist suggested this as the way to explain the origin of eukaryotic cells, and it was largely ignored until the mid-1960s when Lynn Margulis, a biologist, basically took the thinking of this Russian botanist and expanded upon it and then substantiated it by doing work in microbiology, arguing that basically eukaryotic cells represent the merger of bacterial cells and archaeal cells. And the current thinking is that the host cell was an archaean that basically engulfed other bacterial cells and archaeal cells and established what's called an endosymbiotic relationship with these engulfed cells, where the cells that were engulfed established a permanent residency in the interior of this host cell, and over time the metabolic pathways and operations of the host cell and the endosymbionts become integrated to form a collaborative metabolic environment, and that the endosymbionts experienced genome reduction and as a result became essentially degraded bacterial and archaeal cells in the interior of this host cell, and eventually evolving to give rise to organelles. And the poster child, if you will, for the endosymbiont hypothesis are mitochondria. And mitochondria are believed to have been engulfed by this host cell as an alpha proteobacterium, which is a group of bacteria that many people believe was the source for the endosymbiont that eventually evolved to give rise to mitochondria. And the evidence for mitochondria being derived from, again, alpha proteobacteria through this endosymbiotic relationship is essentially the similarity between mitochondria and alpha proteobacteria. The size of mitochondria and alpha proteobacteria is roughly the same size. They both possess dual membrane systems. Mitochondria have in their inner membrane, which corresponds to the plasma membrane of alpha proteobacteria, a unique phospholipid species called cardiolipin. The only place this is found is in the inner membrane of bacteria and in the inner membrane of mitochondria. The outer membrane of mitochondria have proteins that look as if they were derived from proteins called porins, which are transport proteins in the outer membrane of alpha proteobacteria. There is a circular piece of DNA in the lumen of mitochondria, which is believed to be derived from the circular DNA that constitutes the genome of alpha proteobacteria. And the information processing systems in mitochondria are not like that of the nucleus of eukaryotic cells, but have much more in common with that of alpha proteobacteria. And there's other lines of evidence as well, but this discussion gives the strong sense that given these biochemical and morphological similarities between bacteria and, again, alpha proteobacteria, constitutes the chief evidence for the endosymbiont hypothesis. And on the surface, it appears to be rather compelling evidence, to be clear. And so this is part of the reason why this has become such a cornerstone idea in the evolutionary framework and is considered to be the way to explain the origin of mitochondria from an evolutionary standpoint. And I oftentimes am asked when I'm on university campuses, particularly by students that have had a little bit of biology, what is my reaction to the endosymbiont hypothesis? Because particularly as a creationist, as an intelligent design proponent, because it looks as if the case for the endosymbiont hypothesis is such a strong case. And so this is why Steve McCrae asked me if I thought that mitochondria are bacteria, because in a sense, from an evolutionary standpoint, there would be a continuum between a bacterial endosymbiont as an independent organism and the organelle, a mitochondrial organelle, which would be essentially now an enslaved endosymbiont that has become degraded in its biochemical capabilities, that again has been integrated into the host cell's metabolic operations. Now, Steve brings up another question about the autogenic model for the origin of mitochondria, and hence the origin of eukaryotic cells. And to be fair, I had never heard of this idea till Steve asked me the question and then subsequently sent me a link to a website in which somebody is espousing this particular view. And I actually tried to dig in a little bit to this individual's credentials as a scientist, and I couldn't find anything that gave me any indication as to what their credentials were. And as I looked at the explanation that was being offered for the origin of mitochondria through this autogenic pathway, where he was arguing that the mitochondria are derived as kind of blebs from the endoplasmic reticulum, I found the explanation to be extremely superficial, lacking a lot of scientific substantiation, and didn't find it to be compelling whatsoever. And so even though I'm skeptical about the endosymbiont hypothesis as an old earth creationist and as an intelligent design proponent, the fact of the matter is I didn't find the autogenic model compelling whatsoever. And so if I would embrace an evolutionary perspective, it seems to me that the strongest case to date would be the endosymbiont hypothesis. But one of the things that's interesting on this website is that this individual who's proposing this autogenic model for the origin of mitochondria offers a critique of the endosymbiont hypothesis. And the fact that you see somebody suggesting kind of an alternative evolutionary model for the origin of a mitochondria and hence the origin of eukaryotic cells is an indication that even though this is the prevailing explanation from an evolutionary standpoint, it is not without criticism. It is not without its deficiencies. And this is one of the reasons why I'm skeptical that this actually is ultimately the explanation for the origin of mitochondria. I think there are some significant issues with the endosymbiont hypothesis. And one of those issues that I've written about, and I'll post a link to this article in the show notes, has to do with the origin of the protein synthetic pathways that have to be developed in order for mitochondria to function as an organelle. Because the proteins that are found in the mitochondria, at least most of the proteins, have to be produced in the cytoplasm, then targeted to the mitochondria, and then imported into the mitochondria, and then targeted within the mitochondria to either the lumen, the inner membrane, or the inner membrane space, or even the outer membrane. And so this pathway and this process is an incredibly elaborate process that is formed from an ensemble of irreducibly complex systems that then integrate into the queen mother of irreducibly complex systems. And even though I'm not a big fan of the irreducibly, or the argument from design for sort of utilizing the concept of irreducible complexity, in this particular case, I find it very difficult to envision an evolutionary explanation for this level of irreducible complexity, and in fact, don't feel like mechanisms like co-option are adequate to account for the processes that are needed to support protein biogenesis for mitochondria. And so this is one of the reasons why I'm skeptical that this is, in my mind, a problem. And in fact, in the article, I cite work done by an evolutionary biologist who discusses the problems associated with protein biogenesis in a mitochondrial context. And he basically says this is a very difficult problem, and somehow protein biogenesis emerged through an evolutionary process, which in my mind isn't a very compelling evolutionary model. But it's not just me as an old earth creationist and an intelligent design proponent that has expressed skepticism about the capacity of the endosymbiont hypothesis to account for the origin of mitochondria and eukaryotic cells. Just a few weeks ago, there was a paper published in a journal called Biology Direct, in which two evolutionary biologists that are members of the Hungarian Academy of Sciences, one of them is Eros Zathmari, who is a very famous evolutionary biologist, wrote a critical review basically arguing there is no compelling evolutionary model for the origin of eukaryotic cells and mitochondria that would umbrella under the endosymbiont hypothesis that currently has been proposed. And so it's not, again, just me as an old earth creationist and an intelligent design proponent that has expressed skepticism about this idea. Even evolutionary biologists have expressed skepticism as well. And what these two evolutionary biologists point out is that there have been over 20 different models proposed to explain the origin of mitochondria, again, underneath the umbrella of the endosymbiont hypothesis, and they argue that only about 8 of them are even reasonable. And of those 8, they argue that none of them can actually answer a series of 12 questions that they developed that they would argue every successful endosymbiont hypothesis or model has to answer. And they point out that none of the 8 actually can answer all of those questions. And they basically call for a new model for the origin of mitochondria and the endosymbiont hypothesis. In a couple of days, I'll have an article published on my blog, The Cell's Design, where I detail this work, and I give a link to the article, again, published in Biology Direct, if people want to dig a little bit deeper. But for the sake of this broadcast, what I would like to do is identify some of the questions that these researchers believe to be outstanding questions that, again, highlight the deficiency of the endosymbiont hypothesis as an evolutionary explanation for the origin of eukaryotic cells and mitochondria. One of the points that they make is this, that there is no evidence for intermediate forms documenting a transition from prokaryotic cells to eukaryotic cells. They would argue that if this endosymbiont model is indeed valid, that we should actually see single-celled organisms that would be documenting this transition where they represent intermediate grades between a prokaryotic cell and a eukaryotic cell. What they argue is every eukaryotic cell that exists in nature is a fully formed eukaryotic cell that has all of the hallmark characteristics of a eukarya. That there are eukaryotic cells, they do point out, that lack mitochondria, for example, but all of these cells look as if they lost the mitochondria as a secondary event in their natural history. And the fact of the matter is that when you look at eukaryotic cells, again, they all display the hallmark characteristics, meaning that the origin of eukaryotic cells must have happened incredibly rapidly over a very short period of time and resulted in an explosive diversification of eukaryotic cell types. In fact, this has been dubbed by some biologists as the eukaryotic Big Bang and currently lacks any kind of explanation from an evolutionary standpoint. Another problem they point out is the chimeric nature of eukaryotic cells. Now, as I mentioned earlier, the information processing systems in mitochondria are distinct from those found in the nucleus of eukaryotic cells. The information processing systems in the nucleus of eukaryotic cells is archaea-like, and the information processing systems in mitochondria are bacteria-like. And it's on this basis that people believe that the host cell that began this process of endosymbiosis and endosymbiogenesis, I should say more appropriately, was a archaean because of that particular feature. So that chimeric property of eukaryotic cells isn't troubling, but what is troubling is the fact that if the host cell was indeed an archaean, why aren't the membranes in eukaryotic cells archaea-like, not bacteria-like? This is an unusual chimeric property that doesn't have an explanation in any endosymbiont model or any model for the endosymbiont hypothesis. In fact, the argument goes that if there's this process of genome reduction that's taking place, where mitochondria are giving up their DNA, and that then genes from the endosymbiont are being incorporated into the host cell, and there's this transition that's taking place, how is it possible that this happens yet without any kind of evidence for this kind of transition taking place with respect to the membrane systems? And in fact, these researchers argue we know of no instance where there was this kind of membrane transition taking place in biological systems whatsoever. So this is a really troubling feature of eukaryotic cells that defies currently an evolutionary explanation. Related to this fact is the observation that the only electron transport machinery that we see and membrane bioenergetics machinery that we see in eukaryotic cells is found in the inner membrane of mitochondria. Because when we look at bacteria and archaea, for these prokaryotic cells, all have their membrane bioenergetics machinery in the inner membrane, the plasma membrane. And if the eukaryotic cell is now derived from an archaean, then we'd expect that the plasma membrane of eukaryotic cells would have membrane bioenergetics machinery, or at least vestiges of that machinery, and we don't see anything like that whatsoever. So again, another problem that these researchers identify. On top of that, they also are troubled by the steps that would be needed to establish this endosymbiont relationship. Presumably, this archaeal cell was able to engulf the bacteria through some kind of process like phagocytosis. where you would have a phagosome that would be formed. Well, if that's the case, then why wasn't the endosymbiont essentially digested away? How did it survive this process of phagocytosis? On top of that, if it did somehow survive, why doesn't a mitochondria have a triple membrane system where the outer membrane would be derived from the phagosome? Again, a significant problem. And let's say this endosymbiont somehow survived that. If the environment of that host cell was conducive to the survivability of the endosymbiont, why wouldn't it just simply replicate out of control, essentially destroying the host cell? So it's mysterious as to how this endosymbiotic relationship was established, and then how the metabolic integration would have taken place. How would things like nucleotide transporters emerge in the membrane system of the mitochondria so that it would give up ATP molecules that it's making, pumping them into the host cell's cytoplasm for the host cell to use? So these are the types of questions that these researchers identified that represent, to me, significant deficiencies in the endosymbiont hypothesis as a way to explain the origin of eukaryotic cells and mitochondria. And again, it's not an old earth creationist or an ID person that's making these points. These are two well-respected evolutionary biologists that are deeply committed to the endosymbiont hypothesis as an explanation for the origin of eukaryotic cells, but yet they point out that we currently don't have an explanation whatsoever. In fact, in the article, they say this, that the integration of mitochondria was a major transition and a hard one. It poses puzzles so complicated that new theories are still being generated 100 years since endosymbiogenesis was first proposed, and 50 years since Lynn Margulis cemented the endosymbiont origin of mitochondria into evolutionary biology. One would expect that by this time there's a consensus about the transition, but far from that, even the most fundamental points are still debated. There's no single theory that can adequately answer all the questions. Furthermore, and more importantly, some answers have turned out to be untenable in light of new results. So this is why I'm skeptical that this is a valid explanation for the origin of mitochondria. Now, just having pointed out problems with the endosymbiont hypothesis isn't sufficient to make the claim that the mitochondria must and eukaryotic cells must reflect the work of a creator, of an intelligent agent. We need to do more work than that, than just simply critique the evolutionary model. We need to give some evidence for, again, the role that a creator must play in bringing about the origin of mitochondria. But one piece of evidence that we could point to would be the eukaryotic Big Bang. If a creator was indeed responsible for bringing eukaryotic cells into existence, they should show up explosively in the geological record. And that's exactly what we seem to see as evidenced by this eukaryotic Big Bang. We would not expect to see transitional forms, and in fact, we don't. In addition to that, we could look at things like protein biogenesis, which I talked about, and note that these are, again, this is an irreducibly complex system, but it's an irreducibly complex system made up of irreducibly complex systems. There's a hierarchy to the irreducible complexity that, to me, is a signature for the work of a creator, because again, when we create as human designers, many of the systems we produce are indeed irreducibly complex. But I would also point out that we can also explain from a design perspective why there's cardiolipin in the inner membrane of mitochondria and alpha proteobacteria. Well, if this is where the energy harvesting pathways are, where electron transport chain, the electron transport chain is, and where the F1F0 ATPase is in both alpha proteobacteria and mitochondria, then we would expect, again, similarities. We would expect common design to be the explanation, not common descent, and that the shared features would reflect an archetypical design that exists in the mind of a creator. But for example, we now know that cardiolipin, which is unique to those two membrane systems, actually plays a vital role as a cofactor for the F1, F0 ATPase, and its unique structural properties allow it to function as quote-unquote a lubricant, facilitating the rotation of the rotary portion of the F0 component of the F1, F0 ATPase. In addition to that, we now understand why there would be a genome in mitochondria. It turns out that this mitochondrial genome codes for proteins that are highly hydrophobic, which means that it would be impossible to target these highly hydrophobic proteins to the mitochondria because the machinery in the cell is not sufficient to distinguish between the highly hydrophobic proteins that would go to the mitochondria and those that would go to the endoplasmic reticulum. Also, the proteins that are encoded in the mitochondrial genome are critical components in the electron transport chain, and these, by the fact that they are encoded in the mitochondrial genome, indicates that it allows there to be independent control over the production of the electron transport chain, independent of what's happening in the rest of the eukaryotic cell, which is again critical to ensure appropriate amounts of energy production for the cell to use. Also, the fact that the genetic systems are different and the information processing systems are different in the mitochondria and the nucleus of the cell makes sense because it allows for those two systems to be distinguished by the cell's machinery, again exerting independent regulatory control over those two systems. And another thing that's interesting based on some recent work is that the ribosomes found in mitochondria are actually distinct from the ribosomes found in alpha proteobacteria, and that distinction actually, I think, argues against the endosymbiont hypothesis. Also, we note that the genome of mitochondria is enriched in guanine and cytosine, which actually imparts structural stability and chemical stability to the mitochondrial genome. Again, that distinguishes it from bacterial genomes, but makes sense when we think about the nature of the environment inside the lumen of mitochondria. So the point is this, is that if we look at mitochondria as the handiwork of a creator, we see some positive evidences like the eukaryotic Big Bang and the nature of protein biogenesis that points to the work of a creator, and that we can account from a design perspective for those shared features between mitochondria and alpha proteobacteria that many people would point out as being compelling evidence for the endosymbiont hypothesis, in which we're looking at these shared features as not evidence for common descent, but evidence for common design. I'm going to go ahead and bring this broadcast to a close. I've gone on long enough about this. Thank you, Steve, for that really interesting question. I would be interested in any kind of comments that you might have as a follow-up. If any of you want to offer your perspective on the endosymbiont hypothesis or anything that I've said during this broadcast, or just your general perspective on creation evolution issues, please go ahead and do that in the comment section. Even if you don't agree with me, I'm interested in hearing your perspective. Please also take a moment to like the broadcast. This helps us gain visibility in the Facebook feed, and also take a moment to check in and let us know who you are and where you're watching from. I really appreciate knowing who you are and why you're interested in what we're doing here with these Facebook Live sessions. Thank you so much for your interest in what I'm doing at Reasons to Believe. Thank you so much for your support of my ministry and the ministry at Reasons to Believe. We are deeply grateful for your partnership. And I want to leave you with this one final thought. The more that we know about science, the more that we have reasons to believe. I'll see you next time. God bless each and every one of you.